So thank you for joining us. Thank you for making the time, Professor Hornstein and Dr. Genge. Thank you to my colleagues in Montreal that all are on here. A few housekeeping notes. I would please ask you to mute so that we can hear Dr. Genge and Professor Hornstein's presentation. And we will have some time for questioning. And without further ado, Susan Stern, our National Executive Director and CEO. Susan, up to you.
Thank you. It is indeed a privilege. So on behalf of our Weizmann Canada team and the Montreal chapter, led by Joanne Nemiroff, it gives me great pleasure to welcome all of you. Today's event is a dream come true for me. Creating new knowledge and providing hope are pillars of the Weizmann Creed, and this event is a shining example of that. It also happens to be ALS Awareness Month in Canada, so this event is indeed timely. I'd like to begin by acknowledging and thanking some of the vital partners that played an important role in making this critical research initiative between the Montreal Neurological Institute Hospital at McGill University and the Weizmann Institute of Science a reality. Apotex, Inc., CIHR, FRQS, the Israel Ministry of Health, Brain Canada, ALS Canada, ALS Quebec, and the Muscular Dystrophy Canada. We're honored to have representatives from these many partners here today. Welcome and thank you. For those of you unfamiliar with Weizmann, the Weizmann Institute of Science in Israel is one of the world's leading basic science research institutions. The Institute has an over 70-year history at the forefront of discovery, and since 1964, Weizmann Canada has been helping to fuel these critical discoveries. In various well-regarded global rankings, the Weizmann Institute consistently ranks among the best research institutions in the world, and its scientists continue to win awards, including Nobel Prize and Turing Award. World-class excellence is achieved by the freedom to question and investment both in time and resources to find answers. The campus in Israel is characterized by joy and the pursuit of science. The multidisciplinary approach at the Weizmann Institute transcends traditional silos. There's an openness to share knowledge and embrace new ideas. The Weizmann model enables each scientist's freedom to be curious and the possibility of boundless creativity. The model encourages global collaboration. And as I mentioned earlier, today's event is a shining example of that. Creating new knowledge, collaborating with centers of excellence around the world like the MNI, and establishing important partnerships that can help move the research forward, ultimately providing hope to people around the world. Without further ado, I'd like to introduce our distinguished speakers for today's event. Instead of reading their very impressive bios, which you would all have received with the invitation, we asked both for 3 achievements they're most proud of as they relate to today's discussion. And here's what they said. Dr. Angela Genge, Executive Director, Clinical Research Unit, Montreal Neurological Institute Hospital, said, 1, setting up the ALS Center of Excellence at the Montreal Neurological Institute, allowing the ability to run the first patient trials on ALS. 2, developing and expanding the clinical research unit at the Neuro into a unit that manages over 120 trials annually. And 3, receiving the Forbes Norris Award in 2018. Amazing, Angela. Professor Eran Hornstein, Department of Molecular Genetics, Weizmann Institute, said, the discovery of dicer and enoxacin and their relevance for therapy of patients with ALS in 2015, setting up and enrolling to the phase 1b/2a clinical trial, along with the help of Alexandra Sherman, Apotex, and Angela Genge. And 3, Weizmann's forward-looking neuroscience initiative and being appointed head of the new Department of Molecular Neuroscience at Weizmann. You'll get a better understanding of what all that means through their presentations. Ladies and gentlemen, please welcome Dr. Angela Genge, followed by Professor Eran Hornstein.
Thanks, Susan.
Thanks, everyone. I'm delighted to speak to you today. The Weizmann and Weizmann Canada in particular, in fact, Susan Stern, who must take some of the credit for allowing this and helping this project to move forward, has really been central to what it means to be involved in and the research in ALS. ALS is also known as Lou Gehrig's disease or amyotrophic lateral sclerosis or sclerose lateral amyotrophique or Charcot's disease. It has many names around the world, but it all means the same thing. At the time of diagnosis, it means you have 2 to 5 years to live. And from the time you are diagnosed to the time you die, you will relentlessly lose your ability to do things. Because ALS is a disease of motor neurons. Motor neurons die. They start dying in one limb or in control of your voice or swallowing, but ultimately every motor neuron in your body succumbs to the same disease pathway. That pathway is not entirely clear. It may be many pathways. There may be a final common pathway, but the result for the person living and dying of ALS is that they lose strength. The most common things I hear are, my golf game is worse. I can't pick up a pencil. Someone accused me of being drunk and I wasn't drinking. These are the kinds of presentations that a disease so serious in its outcome has. Very benign presentations. Presentations where there's no pain, you don't feel sick. The word sick has a funny connotation because people believe that if you have a condition that is going to kill you, you should at least feel sick. You should lose your appetite. You should feel like you have something like cancer. Well, I'm here to say that ALS is equivalent in its seriousness and its progression to pancreatic cancer and some of the most difficult to treat cancers there are. So what happens with ALS is the motor neurons start to die. They already start before the weakness begins or is noticeable. And it usually affects one arm, one leg, or your speech. And the presentation, because it's so without any other symptoms, can go on for a period of months, up to 18 months. The average length of time it takes doctors to diagnose ALS is at least a year. So if you have something this serious and you lose the first year trying to make the diagnosis, you give us, people like Eran and myself, a very short runway to make a difference. The other thing about this disease is because it can affect different people differently, we have to find things like biomarkers that are universal or universally changed in patients with ALS. Not just use techniques where we just follow one arm or one leg. We don't have the ability to follow the size of a tumor as a biomarker. So to figure out what treatments we're trying that is making a difference is very difficult. We've been left with just assessing people's strength or how they function as a marker of whether or not a new drug is effective. That's very late in the game. What we want to be able to do is, one, diagnose those patients who have ALS within 3 months of them starting the symptoms. And there's a lot of buy-in we need from a lot of people, including patients, to be investigated, to be diagnosed this way. And number two, we need ways of marking the disease that can show a benefit from a drug before we see the difference in how the patient is functioning. So we can make decisions about what drugs are better for patients with ALS and what drugs are worse. So what drugs are not working. So for two very different reasons, the things that Eran has been working on at the Weizmann, it's fundamental to what the whole field is. We get together, we used to get together quite often. We now Zoom together quite often. But the issue is the same. We have to find ways of diagnosing this disease and following this disease so the treatments we come up with can be tested early and quickly to see if they work or whether we have to try something new. The only other thing I'd really like to say, and this is more for the patients and people who around who are worried about ALS, as there are some breakthroughs right now, but they're in the genetic forms of ALS. So we're getting some good breakthroughs in treatments that are happening in the genetic form. So now we test everyone with ALS for a genetic form. So between 10 and 20% of patients have a genetic cause to their ALS. The second thing I'd like to say is just because your tests are normal does not mean you don't have a very serious disease. Because one of the main reasons that patients take so long to come to my clinic, it's not because my secretary doesn't answer the phone, because she does. We book people within 3 weeks of them calling, and most patients within a week. The problem is that doctors follow tests, and they keep doing tests, and in ALS, all those tests are normal. So it's not until you get to a specialized center where they do one test and now possibly two that you get the diagnosis and we can start treating. So with that, I'm going to hand it over to Dr. Hornstein, who is going to fill you in on this wonderful Weizmann sponsored activity.
Yeah, I hope you hear me now. So thank you, Angela. It was very exciting and to the point, and also Susan for the introduction. So I'm going to share with you a story today that I think is a great story at any level, and it has to do with the development of a new therapy for ALS. I'll start at the end. We are right now enrolling patients in a clinical trial that is led by Angela at the Montreal Neurological Institute. And we hope that within a year, we'll have our early insights to whether we have something that is powerful at the end. But I would like to tell you what are the early things early steps that took us into an adventure that is now almost 5 years long. So the work started as basic research in my lab on ALS. So to start, we were having an interest in ALS as a disease, but we did basic research, meaning we wanted to understand what's the underlying mechanism, why the disease happens. And if I can say, I can simplify the ideas about the disease. So of course, if the disease attacks the nerve cells in our brain, so probably much of the events of what happens in the brain and what is derailed probably involves this tissue. And we wanted to understand it in a way that provides insight, because overall the disease is a mystery. We know tiny bits and pieces, tiny early clues about genetics, about genes that may drive the disease, as Angela mentioned, but we don't know what are the main pathways, the main internal events that drive the brain to progressively fail. And through research, we figured out a very important and central mechanism that is probably one of a handful of dominant elements in the disease that fails. It's the failure or insufficiency of an enzyme called dicer. Okay. Now you probably know of many activities inside of our body. For example, enzymes that keep our metabolism, our sugar level, our heart rate is controlled. And basically, dicer is a little machine inside our cell that is critical to each and every cell in our body. And particularly in ALS, in the relevant tissue in the brain, it fails. It is inhibited. And then like a broken car, it cannot do what it should have been doing. Now, from that point on, once we identified it, it was also obvious that if we can recover this broken enzyme, if we can put it on, ignite the engine again, maybe we can provide a candidate therapy that we can explore. And we did exactly that. We looked for small molecules, meaning drugs like the ones we take when we have an headache, that might be directly increasing the activity of this failing molecular machine called dicer. And we found one. The name of this molecule is enoxacin. And funnily enough, we did not need to figure out the chemistry from scratch. It was once, many years ago, used as an antibiotic therapy because it has a property, I would say one property, to kill bacteria, to kill germs when you have infection. But it had another secret property that it could enhance dicer activity. Now I'm telling you about this other property because we were very naive at the time. And once I contacted Angela and said, well, we can consider a way to do a clinical trial because I think the drug is available at the market. We can buy it off the shelf and just go and see if it has an effect in ALS patients. And just imagine if it would work, it would be wonderful. So we started all that is needed to do the trial. We went on to a very competitive grant agency that supported us. And you heard already a long list of super important partners and collaborators, most of them in Canada, through patient organizations and also governmental entities that support our work plan. But the major element there was to just buy the drug off the shelf. And when we tried to do it, we figured out it was, out of order. It was not available anymore, and no one could allow us just to buy it. So all of a sudden, it became a big issue how to move forward. And then I would say two elements came to our help. One was Susan Stern, who was very influential in trying to find a way to help us through. And she introduced us to Barry Sherman, the founder of Apotex. And the Sherman family, first Barry, and later when he passed away, his daughter Alexandra, were very instrumental in allowing us to use their huge resource, their drug company, Apotex, to generate the drug that was essential for us. So we got, after a while, to get our hands wet in working with the pharmacist inside Apotex to redevelop the drug that in the first place we thought was easy to get. Now the drug is available. It went through all the quality controls, and it's ready to use at the Montreal Neurological Institute. And our hope, again, is that we'll be able to say something meaningful about the safety and the potential effect the drug has in ALS patients in roughly 1 year from now. I think the story has additional values that I would like to share with you. So again, many partners at different positions getting engaged in trying to move forward an important biomedical issue, and that's wonderful, and is transatlantic, Israel and Weizmann and many partners in Canada. And I think I could not imagine a better partner than Angela in running the clinical trial. Her system and her team is so professional and effective. It's just wonderful to work with them. I would like to mention that the idea of going from a completely basic science to the bedside, to a clinical trial, is something that is not trivial. At times, it is done by drug companies that buy an idea or license an idea and try to do all the clinical trial from a company point of view. Doing it by two academic institutions is really unique, is really exceptional, and it has the value of us keeping a controlled academic, intellectual, and clear focus on where we go. The last thing that I wanted to mention in context of working at Weizmann in neuroscience, and this may have to do with what Angela mentioned about cancer as a reference. So you know that throughout the 20th century, there was huge investment in understanding from a scientific point of view cancer and the mechanism of what's causing cancer. And for a while in the late 20th century, there was a frustration among physicians and scientists that we do not develop new therapies. So we had a really good understanding and the therapies were not there yet. And now at the 21st century, I would say there is a drug revolution where biological drugs and antibodies and cell-based therapies all get into the cancer field and transform it dramatically and progressively. So every few years you see dramatic change. I believe that neuroscience is walking on the same road with a noticeable delay of, I would say, like 15, 20 years because of the complexity of the brain diseases. But we are on the right track, and basically we'll get there for ALS, for other dementias, and for other brain diseases. It's just a matter of keeping on the science active and then transforming or translating the scientific observations into trials in patients. And Weizmann also understood it and initiated as a flagship operation, a new initiative to facilitate and enhance neuroscience on campus. And for this reason, Weizmann now established just last month a new department, the Department of Molecular Neuroscience, and is building a new institute for neuroscience that would bring together cognitive systems neuroscientists, the ones that try to understand how we think, and molecular neuroscientists like myself, the ones that try to understand how the brain and the machine fails and how we can recover it. Yeah, so with this, I think I said most of what I really want to say, and thank you for listening.
Thank you both for these very informative presentations, and we will have a chance for questions from all the participants in a few moments. I also really appreciate the way both of you are able to explain the science in a way that we can easily understand. And that's not trivial and it's not easy, and I do appreciate that on behalf of all of us listening. We don't have a crystal ball. If we did, I'd like for a moment for both of you to dream forward for us. Angela, dream forward. First of all, what could this mean for the treatment of ALS in the future?
Thanks, Susan. Well, I think the big breakthrough will be if we can document the importance of this dicer activity and the microRNAs that Eran has also identified as important markers. And if we can show movement of these two parts of the pathophysiology of ALS. So can we show movement? Because that movement could indicate actually a positive effect of the drug, whether it's an oxytocin or something that's developed from an oxytocin, that's okay too. But if we can see movement, if we can document this as reliable ways of measuring ALS and changes in ALS, then we have something that we just don't have yet. It'll make a huge breakthrough in our ability to test drugs in patients faster and more effectively, and to determine what drugs will work at a much earlier stage in the development of these drugs. A secondary blue sky, as you like to call it, Susan, and we have a blue sky today, so that's kind of nice. Secondary benefit would be, could it be actually a diagnostic marker? Is this something that we could develop that we would be able to test for, that all those people that can't believe they have ALS because they don't feel sick, could we find it, turn this into a diagnostic biomarker that says, yes, you do, we have to just proceed forward, or no, it doesn't look like you do, let's look harder for a mimic of ALS. Wow. That would be game-changing, absolutely game-changing for us. So now Eran gets to dream big.
Yeah, dream big, Eran. Let's hear.
Yeah, so I would say big enough would be that, again, either an oxytocin or a second generation version of the drug would be actually effective in patients. But I would like to come back to the reference I just made a minute ago and say, I think we are on the verge of a way bigger set of waves coming one after the other, where discoveries would be transformed into therapies. And it would not happen only in 2022, but I believe that the next 15 years should be dramatic in brain medicine, and it's diseases that are now incurable and sometimes tragic for patients and families would be things that we can cope with, transform into something that is chronic, more subtle, and not deadly. And that's, I think, the real hope that physicians and scientists have for this field.
And why is the brain referred to as the final frontier?
Oh, I think because of its complexity, right? It's such a challenging machine. There is a logical loop in us using our brain to understand our own brain. There might be, a naughty reference to universal frontiers outside in deep space, but I think it just for sure the most complex machine we know of, and we are, in a sense, we are like engineers that met alien technology that we did not create, and now we try to figure out the rules and what makes it tick.
So the most complex organ and the one we know the least about.
Mm-hmm.
We're going to open it up in a moment to questions. Angela, you're known internationally for the superstar of clinical trials, and people look to you as the gold standard. For those of us who don't know the stages of clinical trials, before we open it up to questions, can you share with us, we've started, I know there are 6 patients so far who have received, who've started. Can you talk to us about the stages of clinical trials? And then I promise everyone will open it up to questions.
Happily. So clinical trials have several stages. We have learned a lot about clinical trials in the public just because of the COVID vaccine and the vaccines that have been developed. It's been really a very positive outcome of a terrible epidemic, that the general public have been educated both on how difficult it is to develop therapies, or in this case, vaccines, but also how these breakthroughs in science can translate into astronomically fast development of effective therapies when everyone works together. So what happens is there's a whole process prior to the first patient receiving anything, in which you look at a drug in terms of its toxicity, in terms of what the levels of the blood are, depending on how you give it. And a lot of that with enoxaparin was done years ago when it was an antibiotic. Now we tend to do it in a slightly different fashion because even in science, so there are changes that happen all the time. We have a very specific way of determining toxicity of new drugs in various people, in various systems. Once we know what the top safe dose is, and we call that the maximum tolerated dose of a drug preclinically, like before it's given to patients, once we know what that is, we can then go and give the drug to patients at a much lower dose, knowing that we're going to try to build up to the highest dose people can take to have an effect, to not have an important side effect. The phase 1 period of drug development is what we call safety and tolerability. So the entire purpose of phase 1 is to make sure that the drug is safe in the people you're giving it. Sometimes these drugs are given to what we call normal healthy volunteers, which is not only grad students and people who work in universities, but actually the man on the street, to check to see if the drug is well tolerated in people. And again, a lot of blood tests are involved because we want to see what the levels are in the blood of any of these drugs.
So everyone, sorry, Angela, everyone in phase 1 gets the drug. There's no placebo involved.
So in the traditional models, there is a placebo, but there are oncology-based models where there is no placebo. And so you don't have to do a placebo in the early studies. There are ways of doing it without a placebo, particularly if you're checking more than one dose.
Great.
And so once you finish phase 1, and depending on how you're doing these studies, you either can go straight into the group of patients that you want to develop the drug for ultimately. And so those are ALS patients. It's still considered phase 1, but they break it down into A and B if they do both parts.
So we're in phase 1B now?
Yes, we're in phase 1B because we're in patients and we're checking a couple of doses, and the official term for it is a safety and tolerability study, because even though we're giving it for 4 weeks, that's 2 weeks longer than enoxaparin was originally developed for, because it was developed as an antibiotic to be used only for 14 days. So then after that is done and you determine what the safe tolerated dose is, then you go into what is called either a phase 2 or a proof of concept. They're interchangeable expressions. When you hear proof of concept, that usually means a big part of the trial is the kind of work that Eran is doing, looking at the biomarkers to see if you're getting a change in the biomarkers with the dose you've chosen in the patients, and also giving the drug for slightly longer, because you need to start seeing patients taking a drug for 3 months or 6 months. Usually phase 2 or proof of concept are only 3 months, but sometimes people take them in ALS as long as 6 months. Once you finish phase 2, you go into phase 3, which is where you make the final determination of whether it's working to change the course of the disease.
In each case, sorry, I keep jumping in, sorry. In each case, you're expanding the group.
So in each phase, you're increasing the number of patients. You may actually be making the group more homogeneous. So you're making the inclusion criteria tighter because you want to study a particular group of patients with ALS. But you're increasing the numbers of patients. You're increasing the numbers of weeks the patients get a drug. You are not necessarily you're definitely not changing the dose at this point. You're doing other things to get yourself to the point to prove that the drug is actually working in the fight against ALS. And then the phase 3 is where you prove that it works. There is a caveat, and it's an important caveat in ALS and other rare diseases. And that is, you are sometimes allowed to use what we call an adaptive design. And this was actually used in the fight against COVID by the people who developed the therapies and by the people who developed the vaccines, because there are 2 groups, right? 2 companies have developed really good therapies to treat people with COVID so they don't go on to have more serious disease. And then 3 of the most famous companies, although there are several others, developed the vaccines to try to prevent COVID. So 2 groups, and they both use adaptive designs to speed up the development. The main purpose of the adaptive design is to not have a gap between phase 2 and phase 3. And you're really, really driven by the science. So you're really driven in this case by the dicer levels and the microRNAs that Eran is going to be validating and using in the later phases, because that allows you to make decisions faster. and ultimately to get an effective drug to the general patient population as soon as possible. So the treatments for genetic diseases that are now in phase in the pivotal trial actually use this adaptive design. So the companies, and they're a couple different companies, but one company in particular started with the phase 1 single ascending dose in patients, and without actually stopping and restarting, they used an adaptive design to just keep adding patients and lengthening the study. And now they're within 2 months of having information on whether or not their drug is working. They had limited biomarkers. What Eran has developed and what we're really working with him on, in addition to it being anoxacin, is making this biomarker so robust that we can get through this program, essentially not having to treat 500 patients, but because of Dicer and the technology that he's working with, we would be able to prove efficacy faster with fewer patients. So that gets you to approval and access much faster. That was a very long answer to a short question. I apologize.
A very comprehensive one. And, you know, we've learned this year, it's been a very long science lesson, and the importance of science literacy. These are things that, you know, knowledge that may be taken for granted in the medical community, but for the rest of us mortals, you know, we need to ask those questions and get our heads around that information. So I promised that I would stop talking and open up for questions. So either unmute yourself and jump right in, or type your question in the Zoom. We have the special privilege of having Angela and Eran with us, so please feel free. Gladys, I know you have a question in there.
I just want to let everyone know, I apologize. I will have to drop off in about 6 minutes. So if you have questions for me, bring them, and then Eran can close it up.
I think that it goes to both of you. As you're looking at the clinical trials, you said that approximately 20% of the patients are genetic. How does this fit in with your biomarker? Do you see the same biomarker in the various genetic patients that you know of?
Well, we don't yet know, Gladys, if Dicer gives any additional clues in the genetic forms. The advantage of the genetic disease, and if you can call this an advantage, is that the gene mutation gives us an opportunity for a very specific biomarker to that gene. So they sort of get a head start on the biomarker because we have something we can create from the gene. Dicer will apply to all patients, we expect, just like another biomarker called neurofilament applies to all patients, including the genetic patients. We expect that Dicer also will, because of the pathway that it's in. But the genetic forms get a head start because we can develop very specific biomarkers to the gene.
Well, I was just asking whether or not you see a correlation between Dicer in the genetic, the genetic disease patients, as well as in the normal patient. Well, not normal, but the other patients.
So when the general notion, the general appreciation in the field of ALS is that while we can have a strong anchor of how the disease starts in some of the patients by looking at the genetics, the final common pathway is probably the same. So whatever happens to those patients that we know about their genetics is molecularly and clinically similar to those patients where we don't know the genetics, with a few exceptions. But as a first approximation, I would say people think that the genetics is really informative, and those patients that we don't know their genetics, they either have a genetics with unknown genes that are yet to be found by ALS geneticists. So that's like, but still the disease converge on the same pathway, and maybe it's the dicer pathway as we suggest, or that it converge elsewhere. But basically, again, if I'll take again a traffic image, so imagine traffic jam is really bad. It can happen because traffic lights break down. It can happen because of weather, but eventually it's a traffic jam, and then the cars do not move. Dicer failure is a downstream event. It's a final common pathway, regardless of the exact genetics that drive the disease.
I think we have, thank you, Eran and Gladys. We have another question for Angela before she leaves, and thank you, Maggie, for this. Will this study include those with Charcot-Marie tooth disease? I'm sorry, I wasn't familiar. Is there a difference or the disease?
Charcot-Marie tooth is a different Charcot disease. Charcot put his name on everything he could. So very special Frenchman a couple centuries ago. Charcot-Marie tooth is a neuropathy, so it is not the same as ALS or amyotrophic lateral sclerosis. But in fact, interestingly, we have a trial starting for Charcot-Marie tooth, which is wonderful because it's another group of patients that have been without therapy forever. So there's also an interest, but it's separate than this. So Charcot-Marie tooth is a neuropathy. Maladie de Charcot is a motor neuron disease.
Thank you. Any other questions? Either unmute yourself and jump in or type it in the chat. And I had a question for Eran and Angela. We're so grateful, and I know you have to jump off soon. So whenever you do, I'm sending you a big hug. And we'll have to get together as we did pre-pandemic over a glass of wine and celebrate this great success and hope. Thanks, Angela.
I hope I'm invited.
Yeah.
Right.
So Eran, I learned a very important lesson when I first started at Weizmann, that when you study at the basic research level, it impacts many diseases. So when you said early on in your presentation, the neurons are dying and dicer, and that affects dicer. Is there any correlation with any other neurodegenerative diseases that could benefit from this new knowledge?
So the simple answer is yes.
Okay.
And we have, I would say, an expected milestones of how we hope to expand. So next sister disease of ALS is called frontotemporal dementia. That's the most common dementia at early age. Then there are the big dementias like Alzheimer's disease and Parkinson's. So I think once we make a strong point in ALS, and I truly hope we will, immediately we'll go to frontotemporal dementia. We believe the molecular mechanism would be the same. Expanding from there has to do with additional considerations. For example, how good and efficacious the therapy would be.
Well, that's exciting to know, because every new knowledge has, each bit of new knowledge has an implication and impact in other areas. Any other questions for Eran about ALS or anything related to this new flagship project?
I'll jump in. Andy Weizmann,
Hey, Andy.
San Francisco. Good to see you, Susan.
Great, you too.
Eran and I go way back to an interesting experience we had in Seattle, which is for another day's discussion, but it's good to see you again.
Thank you, Andy.
Well, now you have us curious, Andy.
Okay, so Eran and I were supposed to meet with this foundation, a representative from this foundation in Seattle, and he had a conference there, and we go, and the meeting was in the same exact hotel where he was staying. So there, so it's one thing is I call Eran before. He's in the hotel, he's gonna hear it. And I meet this guy downstairs, and Eran doesn't show up, and we're having coffee or whatever. He doesn't show up. I call his room, I get no response. And it turns out that he had like the worst flu bug that anybody, and he was completely incapacitated. Like he was wandering around the streets of Seattle looking for drugs to treat this. And while also not being able to really function.
Yeah.
that well. And so he was very
I actually
apologetic.
have a follow-up terrible story to that. So my fever kept on, and on my flight back, I stopped in London and could not wake myself up to the next flight being with such a high fever. And somehow my wife was having this awkward intuition that I will miss the flight. So she would try to call me, and nonetheless I missed the flight. So it was really bad.
Okay, so
but
it's
Andy,
turned its way to the question.
Yeah,
So,
let's
right,
go to the question.
So anyway, we developed this odd relationship over the years, but I haven't seen him in a really long time. So it's good. But I remember from early on that microRNAs were a huge part of your research even back then. So could you talk about microRNAs? You talk about biomarkers now. What are the other implications? Are they biomarkers the primary interest you have now, or are there other things that may be involved as well?
So what are microRNAs? Basically, when we think of how to program a human being or any animal or plant, so we have this DNA blueprint, and this DNA blueprint gives us the instruction of how to build things up. Most of our things of the little machines, such as the one that I mentioned, that are proteins, okay? But like 15 years ago, or almost 20 years ago, a new domain, a new kingdom of molecules came up to be very interesting and really dramatically changed biology. And these are those microRNAs. So they are tiny RNAs. They are super important, super functional, critical for all cells and tissues in our body, relevant to diseases such as the one we talk about now, but they bypass the production of proteins. So they don't generate what would be the expected end products or the expected machines that people thought of until the end of the 20th century as the element that is functional in humans and in animals in general. So I've been studying these, and so they're relevant in ALS, and hence all the clinical trial we have talked about. But these microRNAs are also, because it's so easy to measure them, are probably paving the way to a new field of biomarkers, and might be relevant to, so biomarkers, again, a blood test, for example, a blood test for ALS, but maybe for many other indications. So you can imagine so many things that could have been measured through a blood test if we were having the appropriate one, and now we either cannot tell or need way more sophisticated, expensive means of measurement. For example, just imagine replacing an MRI study or a CAT scan with a blood test, okay? So things like this. And microRNAs are really central in this arena as well. Thanks.
Thank you. Well, with that, and I promised a hard stop. So first of all, I want to thank you all for being here today. As you heard from Angela, this year was a very long science lesson, and learning the importance of science literacy, making decisions for ourselves, our family, our community. We've become a lot more curious, and that's a good thing. Thank you, Eran, for being curious and for leading us and sharing information, providing hope. Thank you to all the partners who made this happen. And for those of you who chose to spend the last 60 minutes of your day with us, we're very grateful. If you're interested in learning more about the new flagship project, or you have a specific area of interest, please reach out to any one of us. You can find our contact information in the email invite or on the Weizmann Canada website. I'm also thrilled that our colleagues from the American Committee joined us, so it's great to see all of you. And my final wish before we close the event is wishing everyone good health and a great start to an enjoyable and hopefully three-dimensional summer. So thank you all and be well.